There was a very good question earlier about stochasticity. We were solving the reaction kinetic models as ordinary differential equations, and there are many sources of noise and stochasticity in biochemical networks. Chemical reactions themselves are stochastic. In particular, most biological molecules, unless perhaps your oxygen, have a lifetime. And the probability of a molecule breaking down is stochastic. It's like radioactive decay. And so that gives rise to intrinsic stochasticity, where each decay is independent. Another is that it's actually scientifically somewhat mysterious how molecules react with each other. There's this idea that if two molecules get close enough, somehow they react. Exactly how that works is rather mysterious, especially if they're complex shapes. But in any case, the probability of a reaction per unit time certainly depends on the concentration of the species, but exactly how the probability of reaction depends on concentration is not obvious. In any case, since the individual molecules have to find each other, and since they're moving in some kind of complex Brownian way, the probability that two molecules get nearby to each other is stochastic. And in particular, they don't only have to be near each other, they have to be aligned properly, which is also stochastic. Exactly what those distributions are is something else. The other thing is, of course, that the outside world is stochastic. You have temperature fluctuations, you're in a liquid, and so on. And so there are all sorts of stochasticity that are involved in biochemical networks. If you actually want to model stochasticity, it winds up, there's a huge literature. There's a nice book out of Los Alamos that talks about various numerical algorithms for simulating stochasticity. The classic algorithm is a Gillespie algorithm, which is about 35 years old now, which essentially says that you're actually going to model all of your molecules as individuals. And so you have a big stack of molecules, and at each time you randomly pick a pair of molecules, throw the dice and say, did they react or not? It winds up that that kind of algorithm first doesn't necessarily represent the stochasticity that actually occurs in biology very well, or in chemistry very well. And secondly, the algorithm gets very slow as the number of molecules goes up. So for ODEs, if it's a 1 millimolar solution, a 1 molar solution, or a 10 molar solution, you don't care because solving the ordinary differential equation is the same. With the Gillespie algorithm, if you have 10 times more molecules, it takes 10 times more time to run. And so that's great if you're in picomolar concentrations. Gillespie's fine, but you sure don't want to do the Gillespie algorithm for oxygen. And so there are now much fancier algorithms that combine ODE and PDE methods and optimize the stochasticity. Those are not things that are available here. They're not things that, as far as I know, are available in most solvers. They're still research areas about how to do optimization. Gillespie is the classic stochastic method, and it's available. In tellurium, once you have your model specified, you could directly simulate the same network that you've just written with Gillespie. And you do that in a very simple way. Instead of saying result equals r dot simulate, you do result equals r dot Gillespie. And then you have the start time and the end time, just the way you do for your ODE solver. And then if you want, you could specify the time step. In general, because Gillespie, each time step is a random amount of time, it doesn't line up the time steps for the outputs. That could be an issue when you're doing things like plots. And so using a time step forces the output to be aligned with a fixed amount of time. The other thing is that typically in our simulations we use concentrations, and often we normalize the concentrations between 0 and 1. Gillespie, actually, the number really is the number of molecules. And so you have to scale your simulations up so that if you say 100, that means 100 molecules, not 100 molar. And so if you're going to convert from ODEs to Gillespie, you have to remember there's a unit conversion. And that's not an option. You have to do it. Okay. So let's do a very, very simple, go back to our super simple equation. This was, you could use, my example here is what you could use, but you could use the one you just did with the cascade reaction. If you save that, you've got your cascade reaction, you could use that. The key thing is that whatever your initial concentration, remember, I don't remember, we used 10, wasn't it, for A originally? Okay, so you probably want to make that bigger. Making it, say, 1,000 isn't really going to change anything. It'll just scale. It will, as far as the OD is concerned, everything will look the same, except the scale of your y-axis will be different. So why don't you try running your stochastic solver, I mean your cascade model that we just ran. Run it with the R equals 1 case, the ratio equals 1 case, but set the initial amount of A to be 1,000 rather than 10, okay? And everybody get that back on their screen, right? Originally, these slides were further down in the lecture, so they reference a few things that we haven't gone over in detail, but they're not complicated. So does everybody have that old simulation running from earlier today? Fine. So we were using the plot function in Tellurium, but if you want to overlay plots, it's better to use this plot array. So instead of typing plot, type te.plot array, parenthesis result, and try that and make sure that it works. I mean, really, the specific example I have here isn't important. All that matters is the initial number of molecules and the solver. As I mentioned, if you run simulate and then simulate and then simulate, it keeps taking you time forward. If you want to reset the value so you start over again, use r.result. r.reset. And there are a couple of options, but r.reset all, open, close parenthesis, sets the simulation back where it started. And then you could do result equals r.Gillespie from whatever time to start and stop time you used before. And then you could do te.plot array again. And depending exactly on how your ID is configured, you may either get a separate plot window or it'll actually plot it on top of the first plot. And that, unfortunately, is a question of how your environment's configured, not a problem, not something internal to Tellurium. Okay. But the key thing is to do the stochastic simulation, you just replace the word simulate with Gillespie. Because of the way plotting is done in Python, the IDE will handle plots somewhat differently. And so for example, Spider will by default put these things on the same plot. But some IDs by default will put them in different plot windows. And that's a setting in the environment you're using rather than in the. But at worst, you should get two separate plots, one of which has one and one of which has the other. So you get something, you're going to have more lines because you've got five chemical species, but it should look like that. Okay, so what you should see here is that they're not identical, but the the Gillespie line is a little bit wiggly, but it basically follows the ODE curve. And from the point of view of the kind of simulations we do, if you're off by 3 or 4%, you don't care. It means as far as you're concerned, if you have 1,000 molecules, you might as well use the ODE. And that's good to know. One of the problems is, what do you do if sometimes you have 1,000 molecules and sometimes you have one molecule of the same species? And that's where these fancy methods, which do ODE when you have a lot of it and Gillespie when you have a few, and automatically switch, work, but we don't have that. Okay, so now try 50. So set the initial concentration to 50 instead of 1,000 and run it again. And at least in my case, the overall shape of the curves is still pretty much the same. The time of the maximum is more or less the same. The decay is more or less the same. But the fluctuations are getting pretty big. And so at 50 molecules, maybe I could get away with the ODE approximation, but it's beginning to get not as accurate. And now try it with 10 molecules. And in this case, when I ran it for 10, my A concentration looks pretty good for this simulation, but look at my B. My B concentration is almost uniformly 50%, 70% bigger than the ODE result. And of course, if you really only had 5 molecules or 2 molecules, then it would be much worse. Sure. It's a stochastic model, so every time you run it, you'll get a different result. And so, in fact, that was the exercise, which is, now if you imagine, right now you run it 10 times, and here I've overlaid 10 different runs, you see that they all have the basic result, which is A decays and goes to 0, B comes up and goes down again. But if I ask when did the peak happen, there's a lot of jitter on it. And if I look at the width of the concentration, the standard deviation of the concentration at any given time is 50%, 75% of the value of the concentration. So the answer is, if I have 20 molecules or so, and I think that the time at which things happen or the concentration at which things happen is important, if I were triggering off a level, for example, a fluctuation of plus minus 50% is a big level. There's a big difference. If I cared about the time at which this reached its maximum, I'd have big fluctuations. And so in these sorts of situations, if I asked, at what time does B get to value of 5, I get a huge different answer for the stochastic simulation. And so there isn't a perfect answer to the question, is stochasticity important in these cases? It's going to depend on the questions you're asking, and it's going to depend on the concentrations. But anytime the number of molecules you're evaluating is less than 100, you have to think, you at least have to ask the question, is the stochasticity important? And then once you've asked that question, you have to ask, what aspects of the problem do I care about? And in particular, if I have something thresholded, so it matters exactly what the level is, or if there's something that depends sensitively on when something happens. In both of those cases, stochasticity is likely to be very important. And so I can't walk you through all of the issues of stochasticity, but I do want to give you at least a heads up about when you might want to worry about it and when you don't. Yes. So, Javier says, well, if we averaged over all these time series, would we recover the ODE result? And in this particular case, because the reactions are linear, each run is independent. And so doing this 10 times is basically the same as doing it on a population of 200 elements. If we had nonlinear interactions, or if we were doing something on the basis of this, then in fact, it doesn't average. And there are certain specific examples of things famously things called stochastic resonance. And I normally talk about stochasticity later on. When one of the big things that it's in a sense is rather mysterious in biology is what differentiation is, what cell differentiation is. And we will very often write signaling networks or regulatory networks that we will assume represent differentiation states of cells. Because of stochasticity, the states of differentiation networks have what are called chatter. That is, they will randomly switch between different states. In general, that is not observed to happen much in real cells. When it does, it's usually pathological, like cancer related. And what that means is that the molecular switching that we typically model as primary in differentiation is not all that's going on. And in fact, we know that's the case because you may be expressing OCT4 or SOC6 or something else. But we also know that when cells differentiate, they methylate their DNA, they acetylate and methylate their histones, they reorganize their DNA. And so they actually do all sorts of things to lock in those differentiation states. Typically, when we build simulations, we don't model that. And again, this is mainly a scientific problem. We don't really understand how that information is translated from, say, the expression of SOX4 to the methylation of particular regions of DNA. We know it occurs, we know it's functional, but we don't know enough about how it works to be able to model it in detail. And so that is something we should be aware of and think about, especially when we're talking about differentiation. And if we had more time, one of the things that I would do would be to walk you through, maybe we'll have time to do a little bit of it, a classic differentiation latch, what's called a latch, where you have feedback, cross-inhibitory feedback. And that's a classic model because in the ODE case, it's either in one state or the other, period. When you Gillespie it for reasonable parameter values, it switches back and forth stochastically. It's called chatter, stochastic chatter. And you could ask the question, what's the typical lifetime in the up state and the down state? And the answer is that it's too short. It can't possibly be enough to explain what's seen in experiment. That particular circuit is very important in early embryology. When you have the very first cleavages of an embryo, of a eukaryotic, of a mammalian embryo, I think not Drosophila do everything differently, but in a mammalian embryo, the cells that are going to give rise to the embryo proper and the cells that are going to give rise to the extraembryonic structures have a SOX-OCT latch. And we know that there actually is some cross-switching early on. There's some noise that switches you back and forth stochastically. And then after a few hours, that's locked in. There's some secondary mechanism that permanently sets you into one of those two states. And it's actually been shown that stochastic switching early on is important. But similarly, if that stochastic switching continued, you'd have problems because then you'd lose the identity of the cells that are going to form the embryo. That's specifically the SOX-OCT model is a symmetry breaking model. So if you start with equal concentrations of SOX and OCT with some small stochastic chatter on them, you'll randomly switch it to one of the high SOX or low SOX state. You sit there. That's right. There's a signal from contact that biases you. And what Julio shows you, delta notch. Delta notch is exactly a symmetry breaking problem, where all the cells initially start out essentially the same, You're going to wind up with high delta cells surrounded by notch cells. But which cells go high is a stochastic process. And you'll see when you run it that for the first 1,000 Monte Carlo steps, 1,200 Monte Carlo steps, not much happens because everybody's almost the same. And then small fluctuations mean you get a little bit higher delta, and then the feedback, the positive feedback inside the cell with the negative feedback to the neighbors means you get these spikes. And so so nothing much happens, and then very rapidly you switch to high and low state. So you have an intermediate state that's metastable. You sit there and fluctuate around that, and then you spread. Once you do that, you lock it in.